Welcome everybody to the September 16th meeting of the IMAG MSM Working Group on Multi-Scale Modeling and Viral Pandemics. We have two seminar speakers lined up for today. They've both generously agreed to stay afterwards for conversation. So we will have the opportunity to see what they want to tell us in addition to their short talks. Appreciate their being willing to do that. I need to remind you that the meeting is currently being live streamed on YouTube, and it's recorded, and those recordings will be archived and made available. As always, Reinhardt, Labenbacher, and I are the co-leads of the working group. Jim Sluka, who's on the call, is part of the administration and helps in all sorts of ways. And then people will certainly have been in contact with Bruce Shapiro and know the work that he does as well. Please contact us if you have any suggestions, comments, requests, ideas. We have our usual communications channels. There's the IMAG LinkedIn page. We have our YouTube channels. There's the IMAG Wiki. There is a new feature in the IMAG Wiki. I don't know, Jim, if you want to talk about that at all. And the Twitter and the Slack channels as well. Please do help us get the word out about these, whether it's for people attending the meetings live or for their looking at the archived materials. Are there any announcements that people would like to make before we get started with the seminars? Jim,
Just a reminder that we have a steering group meeting tomorrow morning at 11 a.m.
Anything else that people would like to announce? As a reminder, people are free to announce a variety of things. If you're having a conference, if you're looking for collaborations, if you're trying to hire somebody, requests for help, these are all perfectly acceptable short announcements. And we do encourage you to use this opportunity to let us know what's going on in the community. No. Okay, schedule for upcoming meetings. Actually, Russ Taylor is going to be moved later in the season, so that's going to be changed. That means we do have an open slot, I believe, next week. So if somebody would like to speak up and claim an empty slot next week, that would be fine. We have one speaker so far for the 30th of September. Again, if people have suggestions, those would be appreciated. A couple of empty slots in October. Towards the end of October, we have Jason Shoemaker speaking, and also Cham Hidari going to be speaking. And then we have a number of people scheduled further out in October and November. So please do help us fill the pool. If you have postdocs, students who would like to speak, you would like to speak again, you could speak on not only on completed research, but research plans, community opportunities, software. And if you have suggestions for speakers in particular, if you have suggestions for speakers who would be collecting clinical or experimental data that might be of interest to the modeling community, we'd really appreciate it. Also, if you have connections in industry, whether that be software-related industry, pharma, medical devices, anything that you think where there could be a connection between modeling and experiment, we would very, very much appreciate your input as to who we should invite and who you'd like to hear. If there are people you'd like to hear speak, please put them forward for us, and we will do our best to get them on the roster. Please remember to mute your microphones when the speakers are present. We'll have plenty of time for questions at the end. And normally we go through both talks first before questions, especially since our speakers have both agreed to stay for the discussion session. During the discussion session, I have set up a pair of breakout rooms, which we can use if the discussions become very divergent. So far, we've been generally stayed in one room, but if the speakers would like to be broken out and the audience would like to be broken out, we can do that. I don't want to take any more time away from our speakers. We'll begin with Dan Becker, optimizing predictive models to prioritize viral discovery in zoonotic reservoirs. So please take it away.
All right, great, thanks. Let me just share my screen real quick. Great, yeah, so thank you for the invite to talk today. And so I'm going to talk about a project that I started with a bunch of collaborators last year around the start of the COVID-19 pandemic, focused on how we can develop different statistical models to try to identify likely sources of novel human viruses. And I'll sort of talk about how that work has grown over the past year and a half to give us some novel insights into using these kind of models in a predictive framework. So to start, we've known for quite a while that if you look at emerging infectious diseases broadly, the majority of those pathogens come from animals, and the majority of those pathogens come from wild animals like this fruit bat here. And so it seems really fundamental, but sampling these different wild species is really the first step in identifying which animals are hosts of zoonotic viruses. There's a real fundamental problem, though, in that there are just many, many hosts to sample, and we can't feasibly sample all of them. So even just within bats, there are over 1,400 different species. And so we need sort of some way to zoom in on our different sampling targets. And statistical modeling has kind of come up as one tool to help us prioritize which host species we should focus in for our field sampling efforts. And there's many different statistical model structures we can use, but just from a really straightforward standpoint, we just think about a basic linear model. We are modeling some response that is probably something along the lines of host infection status, whether a certain species has shown evidence of infection with a given virus. And then we are predicting that on the basis of something like different species characteristics or traits, and I'll just call those features here. And this really general approach, whether it's predicting different host species, predicting host virus links, has been really popular over the past decade. There's been studies using a more explicit network approach, some that are really focused on parasite sharing, some that are focused on predicting vector species, predicting host species. There's a wide variety of different models that have been used historically for this problem. One of the issues, though, is that the predictions from these models and the performance of these models are often rarely validated or compared against each other. So we don't really know how these models perform A to B. And sort of systematically, there isn't a clear, let's predict a list of host species, let's go out and validate those. And what that has meant is that we really don't have a great understanding of which models are best suited to predicting likely hosts of these new viruses. And the current pandemic has made it really clear how important that can be. So this is kind of the overarching question that sort of this project is focused around. And so to address this, we sort of leveraged on increasing focus over the past year on betacoronaviruses. So the current virus causing the pandemic, SARS-CoV-2. If you ignore sort of the spillback cases, we really haven't found this virus in any wild species so far. We've found a number of close relatives like rat G13 in rhinolophid bats in particular. And we've known for a little bit longer that coronaviruses in general, and especially the betacoronavirus genus, is very common and very genetically diverse in bats. And so we think that bats are sort of the ancestral host of most of the betacoronaviruses that have spilled over, including the original SARS virus and the virus that caused the MERS coronavirus epidemic. So in early 2020, at the start of the pandemic, I worked with a large team of ecologists, data scientists, virologists to build a predictive model ensemble for prioritizing different host species of the betacoronavirus genus. So we compiled data on evidence of a betacoronavirus infection in bat species. We used a standard bat phylogenetic backbone of a little over 1,100 species. So this is a little bit smaller than the actual bat diversity of 1,400 species, but still hits the majority. And of those 1,100 species, a fairly small fraction have actually shown evidence of infection with betacoronaviruses. To get those data, we did a kind of simple scrape of GenBank. And so this is showing you the bat phylogeny. And all of the purple points are just betacoronavirus infected species. And so I was in some ways surprised to see that you don't really see a strong sort of phylogenetic signal here. Infection is fairly evenly distributed across the bat phylogeny. And so with this large group of people, and all these individuals have sort of used one of their favorite models in the past, we trained 8 different models to differentiate positive and negative species. We used 4 network-based models. So these use information on the position of different species in the bat virus network. And then we also used trait-based models like regression trees, which predict, in this case, a binary response on the basis of different species traits. Each of these models gives us a slightly different outcome. And so we used a proportional rank on the x-axis here, where a lower value means the species is more likely to show an association with the betacoronavirus. And so we found that model performance really kind of varied. So if you look at the ability of these network models to differentiate positive and negative species, some of them do really well, like network model 2, network model 4 to some extent, others perform extremely poorly. Generally, we saw that these trait-based models performed quite a bit better, but again, there was a large variation in model performance. And then we put them all into an aggregate ensemble. And so the overall performance of our ensemble generally was better than most individual models, and did a decent job differentiating positives and negatives. And so this gave us some initial insights into how these models vary in their performance. It also lets us prioritize species that currently have no evidence of a betacoronavirus association. So they're known 0, but they have a very low proportional rank. So they're predicted likely, but unsampled host, really any dot down in that lower left-hand quadrant. And so what we did using some thresholding criteria was identify an initial about 370 bat species that didn't show any evidence for a betacoronavirus association, but based on these predictions, they're likely hosts. And we can kind of map out their distribution a bit, both for these network models and the trait-based models, and get some sort of initial insights into where these certain species might be aggregated, especially when we compare them to the distribution of known betacoronavirus hosts. There's obviously a greater than expected diversity of possible betacoronavirus hosts in the Americas. And we also see a remaining hotspot in Southeast Asia. You'll notice that the observed data, there's also a lot of these betacoronavirus bats in Europe, and we don't really see that as much in the predictions. And so we're not entirely just recapitulating what we see in the data. And so we finished these models in about mid-2020. And so over the past year, what we've then done is systematically track new data on these host species. And so this is part of the VIRENA Consortium, the Viral Emergence Research Initiative, where we've systematically gone through the literature we've gone back through GenBank. We use some transcriptomic methods to basically try to get any new data on these bat hosts. And this process, as of a month ago, identified 47 new bat host species. And then we could compare, how well did our model do in predicting these known species. And so our ensemble correctly identified 36 of these hosts, so 76%, which I think is a pretty good performance metric. And we then did some more systematic comparisons. And so we basically ran what you might be familiar with a receiver operator characteristic curve in a classification problem. So comparing the predicted positivity rate for these models against how many hosts they correctly identified. And so just like in a receiver operator characteristic curve, anything along that middle line is 50%. So the model's doing basically a coin toss. And anything in the upper left-hand quadrant means we're performing better than expected. And you can simplify that a bit by just looking at the average here. And one of the take homes is that the trait-based models, so the blue models, perform very well generally. And so they are sort of farther from that 50-50 line. And they're kind of doing a better job correctly identifying these known hosts, whereas the network models are performing really right along that 50-50 line. So they're doing about random or worse in some cases. And another note is just that the model ensemble that kind of aggregated these results together ends up being more or less comparable to the trait-based models. And so there's sort of two lessons from this. One is that in this case, we were able to use ensembling as a useful method to buffer against mixed model quality. So some of these models performed very kind of poorly, but they kind of washed out in the results compared to the trait-based models. And the other kind of result that is really nice, but in some ways is very obvious, is that if you want to make meaningful predictions about likely host species, this requires including more detailed information on the ecology of the species. So the next thing we did after sort of comparing these models and how well they predicted these known hosts is we just updated them with those 47 new hosts. And this is just to say that we saw an overall increase in model performance. The R-squared went up from about 42% to 62%, so a pretty substantial gain. And this helped us identify, basically revise that number of known hosts, likely hosts, to about 400 sort of suspect host species. So this is just showing you the overall distribution of those 400 candidate hosts. And you can sort of see that compared to some of those previous maps, the shift kind of goes from looking at mainland China down into more of the Malay archipelago. So we're seeing kind of some change in the distribution of risk spatially. There are still kind of some hotspots in the Western hemisphere. And this is also showing you the distribution of those predictions across the bat phylogeny. And so the red clades are the ones identified by a certain algorithm to be especially kind of high risk. And so clade number 7 is the rhinolophid bats, which not really surprising, that's where we found a lot of these close relatives of SARS-CoV-2. But one of the more interesting candidates here is that clade 8 down in the bottom, which is the old world molluskids, especially in sub-Saharan Africa. And these are species that haven't had as many of their members test positive for betacoronaviruses. And so they'd be a really good candidate for sort of field virologists to go look into in more detail. So this kind of entire process showed us that if you want to predict likely host species for new viruses, that whole process would benefit from this dynamic process of making predictions, compiling new data, testing those predictions, and then dynamically updating your models. This is something that we really did in real time as the pandemic has unfolded. And we're now sort of trying to test these predictions again, using a combination of field sampling and also using museum collections, which have had a lot of these tissues preserved and just ready to be sampled. So the last thing I want to mention, just as we wrap up, is sort of what are the next steps for this work in terms of improving model performance. And so something that me and my colleagues talked about last year was thinking more about how we can bring in within host or immunological data into these models. And so most of these models are relying on really coarse ecological traits of bat species, things like body mass, forearm length, their geography, their diet, all really important variables, but they're, in many cases telling us about how they interact with their external environment and not so much about their physiology. And so there's been a real shift in a lot of the disease ecology literature in thinking about how we can represent some of these within host variables. And so we think about this general idea of host competence, the ability to actually transmit a new infection, and that being the product of how susceptible you are, how well you can replicate the pathogen, and then how well you can actually shed the pathogen and have the pathogen survive in that infectious material. And so going back to that model structure, colleagues and I thought about some ways that you can include competence in these predictive models. And so on the one hand, you could use things like challenge experiments or proxies of competence in the field, like isolating Lyme virus. You can actually model that directly as your response variable, but in many cases, that's really challenging. So another option would be to actually take immunological data and use that in your predictor matrix for these trait-based models. And so we've started sort of trying to do that, especially leveraging proteomic data. So last year I worked with biochemist Ben Neely to characterize the serum proteome of vampire bats, which are a common reservoir host for things like rabies virus in Latin America. And using only about 2 microliters of plasma or sera, we were able to identify about a little over 300 different proteins with high enough abundance to detect them. And this graph is just showing you the rank abundance of things like complement proteins, which we know are going to be important for both innate and adaptive immunity. And so a kind of project that we've been brainstorming and something that I think would be really, really interesting here is to do more of a comparative study across a variety of different bat species. This is just showing a small number of bat species that we reliably capture at field sites in Central America, characterizing some of the different immune proteins that are common in the plasma or sera, looking how those vary across these different species. And then importantly, these different proteins, or sort of the Go terms they represent, can become features in these predictive models. And the idea is that that would help us potentially improve model performance even more, because we're actually incorporating some variables on the within host factors that could facilitate or inhibit pathogen replication and shedding. And so that's kind of an active area of research that might be of interest for members of this group to think about. And with that, I just want to thank funding sources, especially members of the RHINA Consortium who helped with this large modeling work. And yeah, again, thank you for your time, and I'm sure I can answer questions during the breakout session after the next talk.
So I assume you want me to share my slide deck, correct? I think you're muted. There you go. I'm doing it.
Go ahead.
Thank you all for taking time out. And I look forward to your questions at the end. And thank you for the organizers for the invitation to speak. This is pretty exciting for a few reasons. So just before I go through science. Oh, excellent. Okay. So applied biomath, and in essence, we're a math modeling company where we have mathematicians, engineers, biologists, pharmacokineticists, software engineers, and we work to help pharma and biotech make better drugs, hopefully make them more quickly, help patients, hopefully save money, maybe not work on drugs that are most likely to be failures. We do a lot of different stuff, but today I'm going to focus primarily on systems modeling. We all know that R&D is very, very expensive. Can take 10, 15 years to get to the market, depending on how you do the calculation and what paper you read. Could be $1 billion, $2 billion. But, if you read the asterisk, that's paying for failures. But, it's not unheard of for a phase 2 to be $2 to $40 million, or a phase 3 to be over $100 million. Like, that's real money per project. And even starting from phase 1, you have a 90% failure rate, give or take. Depending on what paper you read, some of these numbers are a little better, a little worse, but that's about what they are. But this is where I think dynamical systems theory, from an applied mathematics point of view, from an engineering point of view, can really help out. In 2004, the FDA did an analysis, and they were like, just a 10% improvement in predicting failures before clinical trials could save, on average, $100 million in development costs per drug. And moreover, can predictive modeling help maximize the return of investment for companies to focus on first mover advantage, and how do you make best-in-class properties or best-in-class drugs and just being a me too, or someone coming along and taking your market away from you, right? Ideally, you want to kill projects quickly, or the ones that could be winners, how do you accelerate them or hopefully develop them so they are best-in-class therapeutics? And at the end of the day, there's a mantra that you see in a lot of these papers where you want the right drug, the right data, the right patient, the trial design, ... But there's a lot of different papers about that. But I just want to set the stage where we think that systems modeling can really help out with all of these questions. Before I go further, I want to define or use the FDA's definition of model-informed drug discovery and development. But first, they have model-informed drug development. MIDD is defined as the use of exposure-based, PK/PD, biological and statistical models derived from preclinical and clinical data to facilitate decision-making. If you go to their report last year, the annual report from the Office of Translational Sciences, we can see that the FDA is getting more and more of these MIDD projects.
All right, Tom,
Oh, yep.
Tom, did you want to be screen sharing?
I think I am. I thought I was. Thank you
No,
for stopping me.
Your introduction sounded like it didn't need slides, and so
No,
I
but
wasn't
I have slides.
sure.
I'll do that. Perfect. I'm sorry about that. Is this better?
We now have a blank screen.
No, not anymore. How about
There
now?
you go. Now
Perfect.
there you go. There we go.
Thank you. Hopefully you can see I'm embarrassed. Great. So we can see that the FDA is getting more submissions and using more of MIDD in their decision making. But this is only the stuff that the FDA knows about or cares about, right? This is patient safety or efficacy designs or whatnot. But these modeling approaches are definitely increasing in importance. I want to add that these same exact modeling techniques can be done preclinically. And that's where we throw this extra D in for model-informed drug discovery and development. So think of MIDD, but we want to include these mechanistic PK/PD models or quantitative systems pharmacology models. I would also throw in really any kind of math, whether it's bioinformatics, statistical analysis, AI, machine learning, hybrids, to enable decisions before you get into the clinic, as you're getting into the clinic, while you're in the clinic, and even after the clinic. So I'm going to assume that everyone here knows what PK/PD is. PK describes a drug's exposure by characterizing absorption and distribution and stuff like that. It's how, the drug's moving around in the body. PD, or pharmacodynamics, this describes a drug's response in terms of biochemical or molecular interactions or reactions. Here, from this point on, I'm going to talk about mechanistic PK/PD or QSP or QST models. And mechanistic PK/PD, in my opinion, are think of PK/PD models, but on steroids, and they include mechanism. So it's not curve fitting to arbitrary curves and doing allometric scaling, for example, to make predictions, but includes the therapeutic concept mechanism of action. So for example, if you have a bispecific, right, what's the format? How many different binding arms are there? What is the first order K on and K off? What is, how does avidity affect the dynamics? Second order K on and K off. Disease biology, right? How many different types of cells are you hitting? Cell numbers, sites per cell, relevant feedback loops. If it's a gene therapy, how do you impact protein synthesis? You know, whether you're accelerating or decreasing it or shutting it off. These tend to be larger than traditional PK/PD models, but they're fit for purpose. They're made to answer a question or two, to grow with the project. And at the end of the day, this allows one to have one mathematical model that can incorporate several different data types that can provide better preclinical to clinical translation. And like engineering approaches, be more prospective, because you can actually put in parameters into the model that make biological sense. Like going from one indication to another, you might have a hypothesis of how cell numbers change or ligand concentration changes or how turnover rates change. So you can actually make real true predictions in a lot of ways. QSP models, think of those as larger PK/PD models, mechanistic PK/PD models that have a lot more biology, a lot more drugs, but those projects tend to be much larger. Here's an example of what a typical project may look like. There's reactions that we can see in the middle box where this is an example of a bispecific ligand antibody fusion protein where we can dose in a certain drug. It can bind to its ligand, soluble ligand. Maybe this ligand is also an epitope targeting ligand. You can also partner or bind to its efficacy receptor. And these reactions might occur in several different compartments depending on the properties or the macro properties of the model, like numbers of cells and things like that. And the types of data that you typically put in here would be in vitro, in vivo, ex vivo, maybe clinical data, maybe competitor data, comparator data. So you can really try to explore and constrain your parameters and think about simulated patient variability, which is at the end of the day, pretty darn important. So these systems models, you can be done early on when you really have very little data internally, but there might be some information in the public domain. And this is a chance to say, you know, can this even be a drug? Before you start a project, you might only have enough money to, like if you're a small biotech, you have enough money to execute two or three projects, but you might have a list of 10 targets that you want to execute. And a priori, you really don't have any idea which of these are going to be easy or hard to develop. And if you have an idea of desired, let's say, dosing frequency in patients, we can inform these models, even with a lot of uncertainty, and say, you know what, do you need a picomolar binder and dose every 4 weeks? If it's a bispecific, do you need a femtomolar binder for each arm and a 6-week long half-life to maybe inhibit 90%, greater than 90% of your ligand receptor inhibition? If you do, maybe you don't work on that project. Where the projects that are easier to develop, you can at least focus on what's the easiest projects to work on. And then through sensitivity analysis, start to prioritize your most important experiments. Then as the project progresses, you can start thinking about adding more critical downstream biology. You don't want to use too complex models too soon because you don't want to waste the time or money, because time and money and return of investment are critical. But once you've decided to invest in this program and you might spend $2 to $10 million in the next 4 to 5 years, it starts being worth making these more complex models. So now you can think about how you're going to design your lead generation strategy. You can start thinking about your most important experiments, and you work on experiment design and experiment prioritization. And then as you start generating this data, you can now start helping out with clinical candidate selection. And are you in fact better than the competition? If you don't think you're going to be better than the competition, maybe you don't, go into the clinic, because that's where you start losing significant time and money. Or is there a way that you can be better by changing certain drug properties? And then of course you can enable the IND And this is really one of the most important steps to have that better clinical trial design to make sure you're going into the right patients, you're going into the right indication and the right dose or dosing regime, dose and dose frequency, because there's so many trials that have failed because of poor design. And here, when you have that single model that can recapitulate maybe 70 or 80% of all your preclinical data and comparator data that's in the public domain, you will have better human dose predictions. Then, of course, you can continue using this model to make better predictions while in the clinic, and then finally launch. I'm going to have some vignettes to talk about. I don't think I'm going to get through all of them. I'm not going to go deep into the math, but I just want to show you some examples that we have that are in the public domain. So this first project, Sanofi came to us. This is in the public domain, and they were interested in executing or working on a bispecific. And this bispecific was going to have an anchor molecule, target A, but on paper, they had 5 different partners for that bispecific. And these 5 different partners on paper, they looked all equally efficacious. So the question was, out of these 5 different bispecifics, what's going to be the winner or could be the winners, and which ones are definitely the losers? And here are some of the simulations that we can look at to try to answer these questions. And some of the data that goes into the models are like the mechanism of action of the drug, the affinity, stoichiometry, first order half-life. You can put in compartment model compartment volumes for the biology, cell numbers, cell types. But we know a priori what a lot of these parameters are. But once you start looking at combining targets, even with a single monoclonal antibody, you have something that's called target-mediated drug disposition. This is where you bind to the receptor, which you want to do, because you might want to block ligand receptor binding, for instance. But once you bind to that receptor is now internalized, and in that trafficking and recycling process, you can lose your drug. So although you want to hit the receptor, it's starting, which is great, you start to lose your drug. So depending on the affinity or how tight of a binder that is, there's a point where there could be a chance where you're too tight and it's not a good drug anymore. But you can't be too weak because you want to block ligand receptor inhibition. So now that's with a traditional monoclonal antibody where you have one target. Once you have a bispecific, this is a very complex nonlinear system, and target-mediated drug disposition of 1 plus target-mediated drug disposition of 2 does not equal target-mediated drug disposition of the combination. So we really have to think about target engagement, blocking, PK, and dosing using this nonlinear system. So because it is a computer, we can do tons of simulations and then do higher projections down to 2D projections so we can try to understand what's going on. And in these simulations, what we're seeing is repeat dosing for different bispecific pairs where I might have target A, target 1, target A, target 2. The more green, the better, because that means you're getting higher and higher target inhibition. The more red, not so good. And on these axes for each of these simulations, on the x-axis for each of these, we're changing binding affinity for target 1, 2, 3, 4, 5, 5. And on the y-axis, we're changing the affinity for the primary anchor molecule. And we can see right from the get-go, at least from changing these 2 affinities, target 3 and target 4 might be superior to these other 3, right? Would these be easier drugs to test preclinically and bring into the clinic? Most likely. It doesn't mean they're going to be efficacious, but it means they're going to be easier to develop. We can now go back to the project team and say, well, you know, marketing might say you have to do monthly subcutaneous dosing to be competitive. But, you know, now you can start having a rational discussion to say, you know what, if we went to 2 weeks, maybe some of these targets might go from infeasible to feasible. And you can have a scientific, logical discussion with your colleagues to see if you can change the TPP or target project profile. You can think about site of action. So whether it's cancer or RA or whatever, you'll have a disease compartment. And the biological properties like numbers of cells and sites per cell and penetration of the drug are different in the central or blood compartment as opposed to the site of action. And if we think of only site of action, or the central, if we think of only the central compartment, which is this top row, we're looking at similar simulations where in the top, I'm looking at the differences between the central compartment, and the bottom is site of action. And I'm changing affinities for the targets on the X and Y axis. And these red boxes represent a feasible drug. Typically, if you can have a nanomolar binder or tighter to about 1 picomolar, that's pretty feasible to make. And we can see that if I only consider information that you might get from an in vitro assay or ex vivo assay, yeah, this looks like an easy project and I have my design criteria here. However, without thinking mathematically about the actual site of action, this now becomes a much more difficult drug to assess. So you can look at all these drug properties and sensitivity analyses to help out. This next case study was with AbbVie. And here they were interested in developing that bispecific, that picture that you saw earlier, where I had a ligand, which is an endogenous ligand, that if you put into the site of action, the joint with OA, that's really, really good because you're activating the system. However, if this is systemic and it's going everywhere else, you start getting this therapeutic index problem where you're activating these tissues outside of the joint, and you're going to start get other diseases, right? You're tipping the balance one way or another. So to help think about this, they were going to link this endogenous ligand to an antibody so they can have tissue targeting, so they ideally stay in the joint. So you can have efficacy and better safety. So we saw this picture that we were modeling, and the local parameters for this reaction network are different in the different compartments. But just to make a long story short, we can now use this modeling where I might change the targeting epitope expression 1, 10 to 100. I can look at these parameters in the top row in the synovial fluid. In the bottom, I can look at the cartilage. On the x-axis for each of these, I'm looking at the affinity of the efficacy of the drug to the efficacy receptor. On the y-axis, I'm changing the affinity of the drug to its targeting epitope. And I can see that the more yellow, I'm getting better efficacy, and we can look at other tissues for therapeutic index. But we can start thinking about two things. What are the best target pairs that you might want to use before you even start the project? Then once you start the project, what are the optimal drug properties for that lead generation? In the interest of, how much more time do I have?
You can go for another 10 minutes if you want.
Perfect, thank you. So here in this case study, this is an interesting project where the project was stuck in lead optimization. The project is going to be dying soon. And in short, the reason being is this data here, the blue represents the competitor therapy. The 3 green curves represent our partner's best leads. And the mechanism of action of this drug is an antibody that binds to a receptor that blocks ligand receptor binding between 2 different cell types. And if you do this in this autoimmune disorder, the cell that you're trying to hit doesn't proliferate as quickly. So you're getting proliferation inhibition. So that's the goal of this drug. Can we reduce this cell type such that you can be healthy again? And in this assay on the left, we can see that the competitor was far superior. However, in a whole blood ex vivo biomarker stimulation assay, we can see these compounds were equivalent. So long story short, this project was going to die because they were too confused. They were falling behind. They were behind the competitor that was in the clinic. And or they were just going to say, let's find a tighter binder. But what we did is by using fit for purpose modeling, where we don't overpredict, where we make predictions first, we can see that without doing any parameter estimation and just putting in good guesses, I can almost recapitulate the data sets. If I change the affinity of the green curve from a 1 nanomolar to 0.5 nanomolar, that shifts to the left, and that's spot on. I can do something similar with the whole blood experiment using a 10 picomolar and 1 nanomolar binder, and I can almost recapitulate that data. If I change the affinity from 1 nanomolar to 0.5, again, I recapitulate the data. So what that tells us is, we are understanding the data, using the model to do knockdown and overexpression experiments and looking at other possibilities like cell numbers and things like that in your assays. What's going on is this data does make sense. They should have expected this analysis before they did the experiments, because there's another cell type that's acting as a sink. So the question is now, can we predict what's going to happen in humans should this drug go forward? And we did tens of thousands of simulations where this surface represents your average patient, let's say. And what I changed for each of these simulations with the first dose, with repeat dosing, is affinity of the drug, the first order half-life, and then I'm looking how long do I have to I can wait till I do repeat dosing, so I maintain low biomarker number and low cell numbers, because that's how we think the drug works. And what we can see here for the average patient is you're actually a better drug by being weaker binder, right? This plateaus. If you're too tight, essentially I'm binding to this other cell type that's acting as a sink and a vacuum cleaner, sucking everything up. But then by looking at potential patient variability for hundreds of different simulated patients, whether you believe it or not, in every single instance, our partner had a better drug than the competitor. So that would say, don't do affinity maturation, right? But also in every single instance, we were near the peak or on the right-hand side of the peak. So under no circumstance would we have a simulated patients where the drug didn't work. So instead of killing the project, or instead of re-engineering the project, they accelerated this in the clinical development because it's potentially a best-in-class drug. In the clinic, there was additional difficulties. They were going to kill the project again. But what we did at the end of the day is said, don't kill the project. We could recapitulate the extreme nonlinear PK. We could predict ligand receptor inhibition. We could predict the variability in the human dosing from a PK and PD point of view in healthy volunteers. And if you believe this mechanism, if you believe the cyno modeling that we could predict, if you could believe the preclinical assays, let's mechanistically translate this to look at predicting what happens in two different indications. And we can see in this indication number 2, I want these blue lines to be below this red line as long as possible. And if maybe a phase 1B, this drug might look like it works. But over here in a patient in a phase 2 where the drug should work, but they want to dose maybe every 2 weeks or even weekly, this would be a failed drug. So they would have a failed trial. They wouldn't know why. They might have to waste another $20 million or kill it. But what we did is with repeat dosing and simulations and looking at patient variability, we said, you know what? If you could only increase this dose subcutaneously by a wee little bit, you're going to go from a failure to a winner. And the head of medicine said, yeah, sure, let's change the trial protocols. They used the model to do that, worked with the regulatory agencies. Here are some of our predictions. They're not perfect, but this is a true de novo prediction that used no clinical data to make the predictions. Sometimes it's a little high, sometimes it's a little low. But at the end of the day, this project wasn't discontinued. It's potentially best in class. The competitors were discontinued. And this was eventually partnered with a large pharma with an upfront payment of $400 million. So this is an example of where we saved a project early and later. It's going to be a better drug for patients, which is huge. At the end of the day, all this analysis could have been done years before, saving serious time and money and heartache. And I wish they did that. But in the interest of time, I'm just going to skip these last simulations. And the big thing for us is we're hiring. We're looking for biologists and mathematicians and engineers and software developers that want to come and do some exciting science with pharma and biotech. So with that, I will thank you again, and we can open up the presentation to group discussion.
Thank you very much. That was great. Wish we had more time. You have to come back and present some of those other case studies for us. so we can dig in a little bit more. Each one of those is worthy of a full hour.
Yes, they are, or more. Some of our employees compare these to dissertation projects in like 6 months.
Fascinating. So we have time now for discussion. Again, I have breakout rooms put up. Clearly, we had very different topics today. So if people would prefer to use the breakout rooms, we can, but maybe we should start with open questions and then move to the breakouts if necessary. John, please go ahead.
I have a question for each, and we'll start with John Burke. How is the FDA receiving your work? Are there individuals within the FDA organization that you actually deal with all the time? And how are these methodologies being communicated and received by the regulatory authorities?
I can't speak for all of industry. I can only talk about how we've worked with our clients, right? I can say that about 80 of our projects have influenced work with the FDA, whether they were for INDs or clinical support. So about half of our projects, give or take, maybe it's closer to 70, but it's about half in the past 7 years. In every single instance, the FDA has never pushed back on our predictions. They have always accepted our recommendations using the systems modeling. We also do traditional modeling too, right? In all fairness, or our clients do the traditional modeling. At times, because these mechanistic models have mechanism, and you can start testing hypotheses for the FDA, like, what if there's 10% more target? What if there's more cell type? What if the target turnover is different? We can assess, predictions on safety and efficacy. We have at times been able to accelerate phase 1 by 2 to 4 months, which is huge for patients, especially if they have cancer and they're about to die, because you don't want to start at homeopathic doses if you don't want to, right? But financially, that's a big deal for our clients, right? So 100% acceptance thus far. We have never had any negative pushback. At times, we have had, oh, this is interesting. Like, they've actually made comments, right? Oh, this is exciting. And later stage stuff too. Thus far, we have not been asked to directly present to the FDA. Usually our partners do it on our behalf because it's their asset, right? We are ready to come in. We help them practice, right? We get ready for that. But typically our clients do that.
But
now,
very
encouraging. I have one more comment to get your thing. Through the MIDD project, where the FDA is trying to use more of these systems modeling. So they're asking for it. They're pulling this, right? Many of our partners have been asked by the FDA to present our models, but on their behalf. So we're not at these meetings. We, as a small company, was also invited to the FDA to present our modeling philosophy and how we work, which is exciting for a little startup, right? So it is very exciting. And the FDA wants more of this. There's still a lot to be done. There's still a lot of math research. And when I say math, like things like convergence, things like validation, right? There's also modeling questions. There's also high performance. There's still a lot of gold standard research and development to be done. I think of this as the early days of, let's say, pharmacometrics from 30 years ago, right? There's a lot of work still to be done, but it's so far been very well received now. When I started off in this 20 some odd years ago, it was much more difficult.
Yeah, that's your last comment actually answered the other part of the question, is that with the progress that seems to be made on the medical device side with the application of model data in the trials, that is very encouraging that the FDA might seek you out to be a client of yours to help them develop their own understanding of what they would like to see in the clinical trial. That what you're doing applies on both sides very much. So
I think it does. I mean, in all fairness, the FDA has been using PKPD for a long time and pharmacometrics and PBBK. But I think this is exactly why they've started the MIDD program, so they can hear from us, they can hear from large pharma.
yeah.
We have received about $5 million in research grants from either the NIH or FDA. So clearly they're funding this also.
Yeah,
So there is active research for sure.
Okay, and then I'll save my next question to allow other people to insert one before I pose my question to Daniel.
Other questions?
Yes, I have a follow-up question, John. What are the standards for validation that you need to provide to both your customers and the FDA?
It's a new field. A lot of it is, it's not as simple as PKPD, where the parameters in some sense are arbitrary, but you have identifiability, right? They're orthogonal in some parameter fitting space. These models that we work on are quite large. You typically can't get all the data that you would like. So it's a scientific discussion. You're like, look, I can fit the in vitro data, but I'm not overfitting. I might change one or two parameters. I can fit mouse data, right? I predict mouse data, then I can fit it. I predict cyno data, then I might have to change a parameter, just tweak it a little bit. And it's a evidence of weight. But that validation that you just asked and what I alluded to, this is exactly what the field is working on. What do we need as a field to validate the model, if you will? And what do you mean by validate? Like, is it the right model? Do you have the right mathematics? Are you converging? Like, these are very difficult open questions right now that quite a few people are looking at. And Applied Biomath is doing active research in this, right? It's important to us because as we develop the research, we share it or use it, with our predictions.
Thank you.
I wish I had a better answer.
None of us does.
So maybe I can ask a question to Daniel. Fascinating work. I mean, it sounds exceedingly challenging to do that kind of field work. And you pointed out sort of what the genomics of bats were. I was impressed there were so many bat genomes available. I was going to ask though, when you're doing animal sampling to find out if the virus is present or not, can you tell us a little bit more about how you did that? You mentioned museum specimens, as for example, as targets. Do you have any estimate essentially of the likelihood when you say that clearly when you find the virus, it's there, but if you don't find the virus, how certain are you that it's not in the bat population?
Yeah, that's a great question. So, I mean, the traditional approach here would be to, we go out in the field if you were a field biologist, and you would, probably collect swab samples from, oral swabs, rectal swabs from different bats. Depending on the species you're working on and where you're working, you could potentially do some lethal sampling for organs where we know the virus likes to replicate. We could try virus isolation. That hasn't really been very successful, I think, for the coronaviruses so far. So most of the data, pretty much all the data we used for that study was coming from PCR and sequencing. So the initial scrape of GenBank that we did was limited by just anyone who had put their sequences online, and that had actually sort of annotated what those sequences were, right? So it had to say that this belongs to the betacoronavirus genus, and that's not always the case because there's varying degrees of how far you go with the phylogenetics. So I mean, that's from a field perspective. I think what we would do, the museum work is very similar. We have the benefit there of often just very large sample sizes, and often various tissues from the same bat. So that's kind of a low hanging fruit. The question about how do you know if it's there or not there is a great one, and something that, you know, I'm an ecologist, so I'm not a necessarily on the virology side, but you know, a lot of us in the bat community have really been pushing more, at least temporally replicated studies. So for most coronaviruses and most of these bat viruses, we see high seasonality when we look for it. So there's a very short period in which the bats are actually shedding the virus in their urine or in their saliva or in their feces. It often aligns with some kind of reproductive stress. Maybe there's a birth pulse, there's some sort of nutritional stress with a lack of, a very short sort of acute food stressor. And so if we sample the bats, once a year, and that just happens to not be during that period, right, we're going to have this kind of false inference that that species doesn't have X virus. And so, good sort of temporal sampling is a really important way to make sure that we are getting reliable data. And part of the way we can use these initial models is to say, what are the 10 species in the country where I work that I want to sample a couple times a year, and then we can focus on those species and try to test, are we just missing it because we're not sampling them at the right time, or are we, once we start doing more robust sampling, can we get better inference? There's also plenty of people using serological methods to pick up antibodies. Those will be longer lasting. We just restricted our study to just PCR positivity, just so that we kind of got away from any issues of cross-reactivity and that sort of thing. So just focusing on the bats that are currently infected. But that would be another option of comparing those two different data streams.
Thank you. That was great. So how much work has been done essentially going colony to colony to look at variation within a species of viral profiles?
Yeah, so we did a couple of years ago a sort of literature-based characterization of this not for coronaviruses, but for filoviruses in bats, and then the henipavirus, so a subclade of the paramyxoviruses. And we found that, at least within published work, there's kind of a general sparsity of the temporally replicated sampling. So, often what happens is there is a outbreak in humans or domestic animals. There's sort of a rapid sampling regime in the nearby bat community. And then maybe sometimes that's followed up at different locations or different times, but there's not as much of a systematic effort to sample multiple colonies or multiple time points because it's so logistically challenging. There have been a small number of studies that have done this, and you find generally very strong spatial and temporal autocorrelation in your infection data. And that's kind of what we think is often driving these bat virus spillover events, is that sort of just wrong place, wrong time. There's a period in which the bats are shedding, and in a given location, it might be different based on some environmental factor. It just so happens that people or some other animal intermediate host or amplifier host were there, and that sort of facilitates the spillover process. But when we do these studies, we find lots of spatial and temporal signal. The sampling just often not done in a way that lets us really test that. You might go out and sample 2 colonies one year, and then they sample a different colony another year, just because maybe that's responding to a human outbreak, or your logistical resources are short. Sampling is expensive, so there's lots of ways to narrow down what species to sample and where to sample and when to sample, and it makes a lot of inference really challenging.
Okay, thank you. Other questions?
Yes. I'm hoping you're going to tell me you have an incredible freezer full of well-documented bat viruses, and that you have already done the genetic studies to look at mutation. and the character of mutations between geographic populations where, yes, they had a virus. I mean, I think I understand that your prediction is to say that this particular bat colony in this place has a generic coronavirus or something. But the detail of the mutation is part of the question. And the other is, how do you guys do anything with looking at the virus in the context of the bat immunology? And so kind of a simple question is we know bat temperatures change enormously when the bat goes into flight. So the bat temperature goes from 97 hanging around a cave to 107 in the air. They carry coronavirus. The coronavirus does not succumb to the 107-degree bat temperature. What does that tell us about how you fight the next coronavirus, where you can't expect body temperature to ever go high enough to protect you? So questions in that category, your comments.
Yeah, those are very good questions. So from the first side, so the modeling study here was really a species level assessment. So we're predicting different species that might be hosts of betacoronaviruses, and because the way the data are available, we were really limited in looking at just betacoronaviruses as a entire genus. So there really isn't enough positive data on the sarbecoviruses, the subgenus where you get SARS-CoV and SARS-CoV-2, to actually do that kind of study at a finer genetic scale of the virus. And the hope is that as people dig into their freezers more and do more genetic characterization of these viruses, right, that eventually we will have sort of finer scale viral data to train the same sort of modeling approach on a more specific set of viruses. But for now, we were limited in just this general genus. And so, we're including not just the SARS-like viruses, but the morbicoviruses like MERS-CoV and a bunch of other things. But in theory, you could sort of take this approach to whatever, if there are systems where you have, enough positives at a finer genetic scale to do more explicit predictions.
So who do you need to be able to collaborate with? You guys apparently have developed a way of finding the viruses in the animals. Okay, you have predictions, you know where to go look with a high likelihood, better than chance, that you're going to find another mutation of a coronavirus in a bunch of bats in Africa versus the ones in Australia. So in order to get on with this idea of understanding the bounds of successful mutation, which will define the range of potential danger to humans, when do you and who get funded so that the stuff you collect can be studied by the people who have the resources, the tools to study the characteristics and mutations? And how do you connect with who's going to do the immunology part of that triangle? We gotta find somebody that's gonna fund that work.
So I mean,
and I'm done.
No, so I mean the silver lining to a lot of this has been that there is so much more interest in bats and their viruses and bat immunology. There are now a lot of different groups starting to work on this. There are people looking into their freezers for different bat samples. We've been reaching out to at least one museum to start looking through their museum specimens, and then doing, right, if we find any positives, we can do all the genetic characterization of those viruses. We have field partners in different places, right, that either have their own collections of bat samples. I have collections of neotropical bats. I do a lot of my work in Belize. And so there's a lot of kind of stuff happening and funding resources that are, I think, going to be leveraged towards doing a lot of the basic characterization and a lot of the immunology. And so that's one of the real outstanding questions is, how do bats tolerate these kind of viruses? There have been lots of ideas about flight and temperature. There hasn't been a lot of strong comparative support for those yet. And so there's people who are going to be testing that. We're really interested in the proteomic side of things, looking for protein signatures of different viral infections. And that's something that we are actively pursuing. There are other teams looking more at the immunological signatures through different bat genomes. And there are whole groups that are trying to characterize and develop annotated genomes for lots of different bat species. So I think the interest is there, and there's more funding going into that. Recently there was an NIH grant for, there was just a recent NIH call for bat immunology R21 grants. So there are a lot of people thinking about it.
Did you ever see any evidence where the bat was adversely affected by the virus? Either evidence where a bat cluster had been killed because the virus mutation took them out, that it exceeded their immunological capacity. And I would assume that if that happens, we don't have to worry about it, because if it kills off the host, it ain't going to spread. But did you find any evidence of bat disease?
So it's another good question. So, the kind of assumption is often that bats harbor these viruses without any negative impacts. And there's two ways of testing that, right? You can experimentally challenge bats and see what happens. And people have done that for many different viruses, and a lot of them do nothing. Or you can go out in the field and you can mark bats with wing bands, and you can actually test for whether infection has an effect on survival. I'll say that hasn't been done as often as we would have liked, and so we have a really limited understanding of, do things like coronaviruses have negative effects on bats? The one example sort of counter to all of this is rabies viruses, right? Where bats also get rabies, they can spread rabies, but if they're actively infected with the virus, they are going to die from the virus like anything else. And so, that's kind of a case where the virus does kill off infected hosts, but there are other ways that those viruses are maintained in bat populations, and that's another thing that we can only learn more about by doing these kind of long-term field studies. So, for rabies, we think that lots of mixing between bat colonies helps the virus sort of persist. Hibernation might help the virus kind of lurk for a bit and then reemerge. And so I guess it's complicated, and we really need to do either more of the experimental work or do more of this long-term mark recaptured work to actually, learn some of these answers about, do these viruses harm the host or not? Because a lot of the experimental work has been done in so few bat species that, we're not really sure if that's a conserved property across the order, or if it kind of varies between families or other traits of these flying mammals.
Thank you.
Other questions? I certainly, John, I found your talk, again, it was, I wish you had had more time to speak, because there's so much that I would have liked to learn about. Many of the examples you showed were on antibody therapies. Are antibodies, is that because that they're fashionable, or because they're particularly susceptible to the kinds of modeling you do, or just by accident those were the ones you were allowed to talk about?
By accident, they're the ones we're allowed to talk about. They're also, in some ways, they can be easier to talk about also. Like we recently had a publication for an engineered cell therapy with where we did the math modeling and help with the design and stuff like that. For sickle cell disease, engineered platelets, and that was with CSL Behring, right? So like we do cell therapies. We do cell therapies you've heard of, cell therapies that you haven't. We do ADCs, we do small molecules, we do conditionally active biologics, we've done CRISPR-Cas9, RNAis, engineered message. We have a publication with Alexion on lipid nanoparticle delivery or nanolipid particle delivery, however you want to say it, delivery of engineered message. Like it, in fact, the harder the project, I think the more you need this kind of modeling. Because how do you do allometric scaling of some of these mechanisms? We have no idea, right? How do you, it's the biology in PD, like if you look at engineered message or lipid nanoparticle, I mean, the drug is gone instantly, but yet they affect cells. So you have to model the downstream biology. You have to, in my opinion.
Other comment. Thank you. I was going to say, I need to dig through your papers and see if I can find any that are simple enough. I can take them to my undergraduate class, because we have the students do paper replication, and they're doing, well, really reaction kinetic modeling, but of course pharmacokinetic models have the same structure, so they can do that as well.
Fundamentally, a philosophy that we have, so in the Wayback Machine in math, my thing was obviously dynamical systems theory and singular perturbation theory. It's like everyone wants to do model reduction, wants, right? Which is great. And randomly perturbed differential equations. But I became of the opinion that, these reactions matter. If sometimes you have to put a Hill function in, sometimes you have to, right? But when you translate from one species to another, how do you change that Hill coefficient? What does it mean? Like what does a Hill coefficient of 51 mean? And what's the difference between 50? I mean, and essentially the difference is, well, if you're looking at the old gene therapy stuff, you have a degradation term, and when you look at the quasi-static state analysis, you're looking at stability, right? I mean, there's reasons why people do what they do. But I was like, if you're going to do this from an engineering point of view, ideally we want to, and people disagree with me, and that's fine. But ideally, we like to stick to 0 first and second order reactions. These are reactions that you can think about, that you can change experiments for. These parameters mean something, and they're more easily interpretable or interpreted by protein engineers or med chemists or biologists. Like this is what's a sensitive model parameter? Some arbitrary parameter that you don't know what it means, or doubling time. Oh, I understand that, right? It makes sense. And now the computers are getting faster, and it's like, maybe we don't need to do singular perturbation theory all the time, right? Or assume it works and put some function in there that forces you to have some interesting, bifurcation analysis that may or may not be real at all because you haven't started from first principles. So I say that because I think your students could do any of our papers.
As I say, I've got to Google and pull up some, because I really, it's such elegant work, and the results are so beautiful.
They're very, very smart scientists.
Quite inspirational for them. Other comments or questions for either of our speakers? I mean, one question that I kept thinking about was, the two of you come at these problems from such different points of view. One of you is starting from fundamentally genomics, phylogeny, and classic statistical analysis. The other side is coming from this mechanistic, classic mechanistic time modeling. And yet ultimately we have to put those two things together. And so I'm sitting here thinking about how would you connect how would you, for example, starting from the kinds of things, John, you do, and your understanding of patient variability, if you want to understand why do certain bat species have particular virus susceptibility or particular viral carrying, for example, what would we have to do? What if you were going to say to Daniel, these are the experiments you have to do in your 3 bat species to understand why they're different beyond the genetic differences? What would you say?
Well, first, first, I don't know what Don thinks. That's the first thing I'm going to say. The second thing is, I would go back and I'm like, at the end of the day, really what we do is like mathematical biology 101, right? It's like whether you look at this as predator-prey modeling, it's like, can you draw the right model diagram? So I would work with Daniel, like, okay, let's think of this as a dynamic process. Let's put some immune cells in there. I think John asked that question about that. What are the most important things, right? And I would, whether you think of it as a SARS model or whatever, right? And then we can incorporate that gene data. If you assume, and it's a huge assumption, that genes correlate to protein levels, it's an assumption, right? And sometimes it's not true, but at least as a starting point, then that can start inform the models. And we do very similar things in pharma and biotech, where we might have this dynamical system, right? And we'll have loads, our partners will have loads of biomarker data. And they get it because they can, and it's relatively easy and cheap to get the data. But if you do bioinformatics on it, it's like, I can't learn any from this, right? I just can't, right? For whatever reason, or maybe they'll call it machine learning, right? Because why not? But sometimes what we'll do is we'll say, we'll work in tandem. We're like, man, this is a huge disease. Let's look at what's in the public domain, prior knowledge networks, plus the bioinformatics or machine or whatever you want to look at it and say, you know what? Even a simple singular value decomposition. These data sets represent 60% of the variability. Okay, great. That tells us what proteins and pathways to start with. We're not going to explain everything, but at least we don't have the world's largest model with no data, right? Right. So at least we're grounded in reality. Then we can start building the mathematical model, incorporate some other time course data or in vitro data, clinical data, whatnot. But then we can now go to that hybrid approach where we can do tons of simulated patients or sensitivity analysis. And what that does, because now this is linked in some ways to that, let's say, biomarker data, and we can start adding nonlinear data to this other data set. So now we've bootstrapped or enriched the machine learning now, right? Okay, whether you believe this or not, but at least there are these other data sets that could act as hypotheses to put nonlinearity in there, because in the end of the day, a lot of these data points are on the same eigenvector anyway, right? They are, right? Or however you want to think about it, or they cluster together. So this is a way of enriching it. So I could imagine with Daniel, you could do something similar. I'm just not, I don't know enough.
No, I think that's very accurate. So I mean, yeah, I mean what we've been, or at least what I've been thinking, you know, let's say we identify 5-10 bat species, we go out in the field, we start sampling them over a couple different months. We've now kind of built a time series of infection prevalence with enough points that we can then, you know, take a bunch of different variants of SIRS, SIR, whatever variants you want. We can, you know, fit them to the data, compare them with likelihood, whatever fitting procedure we want to do, and start to try to learn more about what is the actual, you know, mechanism by which we, you know, maybe see cyclic dynamics in the bats, we see persistence or not, being able to feed in any kind of immunological data that we have. So yeah, I don't think these things are separate at all. And sort of the point of this initial predictive modeling exercise is really to help us to say, you know, what are the bats we should study in more detail, and then once you've identified those, we can then take a more mechanistic approach to figure out how are they tolerating these viruses, when are they actively shedding them, you know, when are people, or where are people going to be actually at risk of interacting with these, you know, infectious bats. So yeah, I think having that kind of approach would be really nice. We just, we just wrapped up the predictive part. So this is kind of the next step now, is getting people to start doing that kind of sampling, developing those kind of time series, and thinking more dynamically about how these different data streams start to feed together.
I mean, I see Amber Smith on the call, and she does wonderful longitudinal experiments in model organisms. And of course, with rodents, you have a lot. It's a lot easier to do a longitudinal experiment with a rodent than with a rare bat species. How about in vitro work on bats? How many are there cell lines for the main species that you can work with? How easy is it to do that?
There are some. There are groups that we work with that are really interested in developing more cell lines. There aren't a ton right now, so in vitro work is definitely a big possibility. Much easier than maintaining a large colony of bats. There's a handful of bat species that have a captive colony one way or another. They're obviously very expensive. Here with CDC, that is a hurdle, right? And it's a lot of tape to jump through to get a captive colony set up, especially if you're going to be doing any kind of infection work. So I think cell lines are really a really good option for still getting a lot of these data and asking some of these, fundamental molecular questions. So I think that's one area where there's going to be a lot of progress in the next couple of years.
Okay, I've asked a lot of questions, but I find both talks fascinating. Are there anybody want to ask a final question? We're at the end of our period. No.
It's not a question, but
Go ahead.
I do enjoy giving presentations at different universities. I think it's important. Like this, when I was in grad school, this didn't happen, right? It just, I want to help young professionals get jobs, right? And it's hard to know you want to do stuff until you know it's interesting, right? I didn't know I wanted to do this until I was fortunate enough that my postdoc advisor, paid me so little money, and mathematicians demanded more money. And I was like, well, I can't say no. So I asked, can I consult on the side? Like I didn't know what kind of cool research was going on, right? So I go out and evangelize a little bit because I'm greedy. I want to hire smart people, but I also know we can't hire everybody, and I want smart people to go into biotech and pharma. So I do. So if people are interested in that, let me know, right? I mean, I have to do it virtually now, obviously, but I think that's important. And we are hiring. So if students are looking for jobs, go to our website.
Okay, well, that's great. It's good to see that both fields, the bat virology and the quantitative pharmacology, are growing. I think that's nice to feel that you're in a world where things are moving in the direction you believe they should.
Yeah.
Okay. Well, thanks to both of you very much for your wonderful
Thank you very much.
talks, and
Yeah, thank you.
see you again soon.
Bye-bye.